times when we do DNA analysis and we utilize the PCR, the polymerase chain reaction, we are left with a couple of microliters of clear fluid, and we don't know whether or not our results have turned out. So in this case, we need to be able to visualize our DNA. By doing so, we utilize gel electrophoresis as well as capillary sequencing. And this allows us to see the DNA, potentially even in different colors depending on the kind of primers that you use. We put it into a gel with some buffer, and it's charged from one end to the other. DNA is a negatively charged molecule and will travel from the negative end to the positive end. So we always load our DNA in the negative and we watch it travel to the positive to see how far it separates. This allows us to measure how big that piece of DNA was from the polymerase chain reaction. So now we're done running our gel. And just like you would develop film in a dark room, we're going to develop our gel in our little dark room. This is called a UV transilluminator, and it emits UV light, and this allows us to see if the DNA in the gel actually moved and how far. So we're going to put it in this little cabinet, and then we're going to turn on the UV light. The reason we close this cabinet is because UV light will damage your eyes, and that's why you wear sunglasses when you're outside. So now that we've turned on our gel, we can look on the computer because there's a camera in the darkroom that's taking pictures. Now what we can see here is that our DNA is lit up. And this is what the ethidium bromide does for us, is it allows us to see the DNA lit up on the gel just like you would a picture on a film. So what we're looking for is that these populations are the same or different, and how different are they based on their genetics. And we use things called statistics to figure this out. So we tally up all the numbers of differences among different individuals and different populations, and we try to determine if the populations in Mississippi of Kogan grass are different from the populations in Alabama of Kogan grass. Now remember, this is a grass and it doesn't pay attention to state lines.
With the figure like the one above, you can more easily see how the values associated with the DNA of Kogan grass from Mississippi on the left looks different than the Kogan grass in Alabama on the right.
Oftentimes in science, you work in really large sample sizes, so you're able to obtain something called statistical significance. And this is a certain kind of math that allows us to tell whether or not our results are important or not. So now we have several hundred samples, and we have data for those. And we're interested in, why does it grow there? So we're going to ask, how much rainfall does it get? How close is it to a highway? What about what kind of soil type it's on? And also, we have GPS data, which means we know exactly where these plants came from, and we can use Geographic Information Systems, also known as GIS, to try and figure out if there is a correlation or a relationship between the genetic information as well as the spatial data. So where does it grow, and why does it grow there?
Visit the Natural Enquirer website for more information. There you can learn more about how to analyze and understand your data.